Hello everyone, welcome back to SL Biologica. And today I am here with you a new topic, cells of the immune system. So we can discuss the important MCQs and its associated discussions. So this would be helpful for your CSIR, ICMR, ICAR, GPT and GATE preparations for your PhD entrance exam as well as for your KPSC competitive exam preparations. So don't forget to subscribe if you like this video. So let's move to the questions then. So the first question is, hematopoietic stem cells are best defined as. So we have the options. Option A, unipotent progenitors. Option B, self-renewing multipotent cells. Option C, mature blood cells. And D, non-dividing differentiated cells. So what are hematopoietic stem cells? So before moving into that, let's see what is hematopoiesis. So hematopoiesis is the process of blood cell formation from hematopoietic stem cells, that is HSCs, and they are rare self-renewing multipotent cells found in the bone marrow. So these HSCs, hematopoietic stem cells, are rare self-renewing multipotent cells and they are found in the bone marrow, and from HSCs we have the blood cell formation and that process is known as hematopoiesis. And these HSCs sustain all blood lineages. That is why it is known as multipotent cells. That is, it can form or differentiate into various blood cell types. And they maintain their own pool through self-renewal. So they have the capacity of self-renewal. So now let's move back to the options available. We have unipotent progenitor as one option. Then we have self-renewing multipotent cells. So here's the answer, self-renewing multipotent cells. So HSCs are self-renewing multipotent cells. So it's clear from this description itself. So we have the answer as self-renewing multipotent cells. So now let's move to the next question. So the next question is, so before moving into the next question, let's just look into the pictorial representation of hematopoietic pathway. So we have the multipotential hematopoietic stem cell here. You can see this. It could be called as the hemocytoblast. Okay. From there, it can differentiate or it can move into two different lineages. One is the common myeloid pathway and the other one is the common lymphoid pathway. So common myeloid pathway is initiated by the common myeloid progenitor cell, and lymphoid pathway is headed by the common lymphoid progenitor cell. So in effect, this hematopoietic stem cell would be differentiating into two different lineages or different progenitor. One is the myeloid progenitor and the second one is the lymphoid progenitor. Okay. And from myeloid progenitor, we will get the cells which were megakaryocytes, which ultimately give to the thrombocytes. Thrombocytes are actually platelets. Okay. So from megakaryocytes, we will get the thrombocytes. And the other type of cell that is derived from this common myeloid progenitor is the erythrocyte. Erythrocytes are red blood cells. Okay. Then the third type is mast cell. And the fourth type is myeloblast. And from myeloblast, it can differentiate into the various basophils, neutrophils, eosinophils, and monocytes. And monocyte can mature to form the macrophage. Okay, so from myeloid progenitor, we will get all those cells which are thrombocytes, that is platelets, erythrocyte, mast cell, and basophil, neutrophil, eosinophil, and monocytes. Okay, So that is all about the myeloid pathway. From lymphoid progenitor cell, as the name suggests, it will give to the lymphocytes. So we will get the small lymphocytes that eventually forms the T lymphocyte and B lymphocyte. And the second type is the natural killer cell, that is large granular lymphocyte. So from lymphoid progenitor cell, we will get the cells natural killer cell, T lymphocytes and B lymphocytes. And we know that from B lymphocytes, we are getting the plasma cells that is actually giving the antibody, that is actually producing the antibodies. So is that clear now? The hematopoietic pathway? Okay. So now let's move to the next question. So yeah, in humans, the earliest site of hematopoiesis during embryonic development is. So they are asking about the site of hematopoiesis during embryonic development. So let's look into the embryonic development, hematopoiesis during embryonic development. So it actually starts in the embryonic yolk sac, okay, during the first weeks of development, embryonic development, and from there it will then migrate into the fetal liver and spleen, and finally into the bone marrow. So the pathway, it starts from the embryonic yolk sac, then to fetal liver and spleen, and finally into the bone marrow. Okay, so after birth, this hematopoiesis will be restricted to the bone marrow only. So this is how the hematopoiesis is targeted, or its location, its localization is just like that. So let's look to the question back. So in humans, the earliest site of hematopoiesis during embryonic development is. So which one is that? Yeah, which one? Yolk sac. So it's clear. It is the yolk sac. Okay. So let's move to the next question now. The transcription factor essential for erythroid, myeloid, and lymphoid development is. We have the options ICARs, PU1, GATA2, and OCT2. So actually, GATA2, it regulates multiple hematopoietic lineages. So here they are asking about the transcription factor, which is essential for the erythroid, myeloid, and lymphoid development. So we have all the two types of lineages here. So actually, GATA2 is the one which is regulating the multiple hematopoietic lineages. So once knockout is happening with the GATA2 transcription factor gene, then it will be lethal to the individual. So the answer here is option C, that is GATA2. Okay, clear? So let's move to the next question. Icaros knockout mice fails to develop. So they are again asking for the another type of transcription factor, that is Icaros. So Icaros knockout mice, that means in those mice which are having this Icaros knockout, that means these genes will be silenced. So they fail to develop which cells? They are asking which type of cells won't develop in Icaros knockout mice. So we have the options as erythrocytes, T, B, and NK cells. NK means natural killer cells, okay? Then neutrophils and platelets. So Icaros actually controls the lymphoid lineage specification. So in the previous question, we already discussed that lymphoid lineage, through lymphoid lineage, we will get the cells. Which one? B lymphocytes, T lymphocytes, and natural killer cells. So once Icaros is damaged or silenced, these type of cells won't develop. So here the option B, T, B, and NK cells, that option is the correct answer. So yeah, option B is the correct answer. So Icaros knockout mice will be failing to develop the T, B, and NK cells. Okay, clear? So now let's move to the next question. Which of the following is not derived from the common myeloid progenitor cell? So this would be an easy question, right? Because we already discussed the pathway, the hematopoietic pathway. So in that thing, we already discussed how the lymphoid lineage is happening and how the myeloid lineage is proceeding. So they are asking which one of the following is not derived from common myeloid progenitor cell. So we know that from myeloid progenitor cells, we will get platelets, then erythrocytes, and then basophils, neutrophils, eosinophils, and monocyte. So we have the options monocyte. Yeah, it is derived from myeloid pathway. Then neutrophil, again, it is derived from the myeloid pathway. Natural killer cell, that one is actually a product of lymphoid lineage, right? So this would be the answer. The option D, megakaryocyte, that is the progenitor for the platelets. Again, a product of the myeloid pathway. So obviously the answer is option C, natural killer cell. So we will look into that. Yeah, here's the description. Natural killer cells along with B and T cells are derived from the common lymphoid progenitor cell. We already discussed about that. Monocytes, neutrophils, and megakaryocytes are all of myeloid lineage. So we have the answer option C, natural killer cell, that is not derived from the common myeloid progenitor cell. An easy question, right? Okay. Now let's look into the next question. Yeah, the cytokine erythropoietin, that is EPO, primarily produced by the kidney, is critical for. Okay, they are asking the function of EPO, that is erythropoietin, which is a cytokine. So before moving into the options, let's look what this EPO do. So erythropoietin is a glycoprotein cytokine produced by the kidney that specifically induces the terminal development and regulates the production of red blood cells or erythrocytes. So we have the answer here. Its function. It induces the terminal development and regulates the production of red blood cells. Okay, now let's look back to the options available. Option A, stimulating B cell differentiation into plasma cells. So it won't be the answer. Then option B is inducing the terminal development of erythrocytes. Yes, one of the function of EPO is inducing the terminal development of erythrocytes. However, we'll just look into the other options also. Option C is promoting the development of all granulocytes. No, it is not the function. Then option D is inhibiting the apoptotic cascade in lymphoid cells. Again, it is not related to the lymphoid lineage. So the correct answer would be option B, inducing the terminal development of erythrocytes. You have just an easy way to remember this thing. The term is erythropoietin. The name is erythropoietin. Erythro means it is something associated with erythrocytes. So in that way, you can just easily remember that erythropoietin is something which is associated with or which is inducing the RBC development. Okay. So this would be again an easy question, right? So don't forget that erythropoietin is associated with the terminal development of erythrocytes. Okay. Now let's move to the next question. A failure in the steady state regulation of hematopoiesis, possibly due to abnormalities in the expression of hematopoietic cytokines or their receptors, is a potential contributor to the development of dash. Okay, so they are asking if something happens to the hematopoietic cytokines, that is, those cytokines which are associated with or which are regulating the hematopoietic development, what happens or what critical condition will arise? Leukemia, chronic anemia, autoimmunity, or viral infection. So they are asking about the lethal condition that is associated with malfunctioning or a failure of the regulation of these cytokines. So let's look into the scenario. So actually, abnormalities in the expression of these hematopoietic cytokines or the receptors to which it binds will be ultimately leading to unregulated cellular proliferation. So unregulated cellular proliferation is what? It is something which is associated with cancer, right? Cancer is the uncontrolled proliferation of cell. We know about that. So once this hematopoietic cytokines are abnormally expressed, it will lead to unregulated cellular proliferation. So it can contribute to development of some kind of leukemia. Leukemia is a kind of blood cancer. Okay. Then failure of apoptosis in hematopoiesis can also result in the survival of abnormal clones. So this will also lead to the development of leukemia. During the regulatory process of hematopoiesis, this apoptosis or programmed cell death is actually happening. So this one is responsible for eliminating the removal or death of the abnormal cells. Okay, so together with the abnormalities that is associated with hematopoietic cytokines, a failure of apoptotic pathway to eliminate the abnormal clones can also result in the leukemic transformation. So from the options, it is clear that option A is the correct one because it is saying the leukemia as option. So that option A is the correct answer. A failure in the steady state regulation of hematopoietic cytokines or their receptors can ultimately lead to the development of leukemia. Now let's move to the next question. Which statement accurately describes the function of cytokines in the immune system? So they are asking about the function of cytokines in the immune system. So before moving into the options, we will just look what the function is. Cytokines are actually protein molecules which can act as immunoregulatory hormones. And this can play important roles in the regulation of immune responses. So they are secreted by various leukocytes, including the T lymphocytes and macrophages. So now the function is clear. They are immunoregulatory hormones, protein molecules, okay? They can play important roles in regulating the immune responses and they are secreted by T lymphocytes and macrophages. So now let's look into the options. Option A is they are membrane-bound receptors for antigen recognition. Is it true? No. They are not membrane-bound receptors. They are actually hormones which are released. So option A could be ruled out. Then option B is they act as immunoregulatory hormones. Yes, they are hormones and they have the potential to regulate the immune response. So option B should be correct, right? But let's look the other options also. Option C, they primarily transport oxygen in the blood. No, they don't have that function. Then option D is they are only secreted by accessory cells like macrophages. So that one is also not correct because it is also secreted by the T lymphocytes. So which one is the correct answer? Option B is the correct answer. They act as immunoregulatory hormones. Okay. Now let's move to the next question. The human marker CD34 plus denotes what? So which is actually CD34 marker? CD represents cluster of differentiation. Actually, the CD34 is observed in the hematopoietic stem cells. So this marker is particularly used for isolating the human hematopoietic stem cells from the clinical samples. So now let's look into the options. The question is the human marker CD34 denotes dash. Option A is terminal neutrophils. Option B is activated macrophages. Option C is HSC enriched populations. Option D is memory T cells. So which one is the answer? Obviously, option C, HSC means hematopoietic stem cells. So HSC enriched populations will be containing the CD34 marker. So the correct answer is option C, HSC enriched populations. So let's look to the 10th question. Skid mice are used for HSC studies because they option A lack T and B lymphocytes, option B overproduce antibodies, option C resist radiation damage, and option D have no macrophages. Skid means severe combined immunodeficiency. So the skid mice will be lacking various immune responses. So they will be lacking the adaptive immunity so that we can do the xenogenic stem cell engraftment. Xenogenic means something related to cells or tissues which is transplanting between individuals of the different species. So if cells or tissues are transferred or transplanted between individuals of different species, then we use the term xenogenic uh, engraftment or xenogenic transfer. So the skid mice usually lack the adaptive immunity, so it allows the xenogenic stem cell engraftment or transplantation. So now let's look back to the options. They lack T and B lymphocytes. Overproduce antibodies? No, it won't overproduce antibodies because they lack the immune response. Resist radiation damage? No. Have no macrophages? Actually, the answer is option A because they lack the adaptive immunity. So they lack T and B lymphocytes. So the correct answer is option A. They skid mice. They lack T and B lymphocytes, and it could be used for the HSC studies, mainly xenogenic stem cell engraftment studies. So now it's clear. Let's move to the next question. Graft versus host disease, that is GVHD. The results from what? So that is the question. So before looking into the options, let's see what is graft versus host disease. What is that? Graft versus host disease. You must have heard about transplantation, right? So it is something which is associated with this transplantation. So a syndrome, this is a syndrome which is characterized by inflammation of different organs. And this is actually associated with bone marrow transplants and stem cell transplants. So this is a disease or a condition where inflammation will be occurring in different organs, and this is a result of bone marrow or stem cell transplantation. So how it is occurring? So they occur when the donor's immune system, okay, they act against the recipient cells. So we have two participants during transplantation. One is the donor and the second one is the recipient. So in GVHD, as the name suggests, it is graft versus host. So graft here is the donor cell. Okay, so here the immune system or the white blood cells of graft or the donor cell, the transplanted tissue, it will act against the host. Host means the recipient system. So we will develop multiple inflammatory reactions in different organs. Okay, so that is GVHD. Don't confuse this term with the transplant rejection. Rejection is actually different. Rejection means we are rejecting the accepted transplant. Okay, here the host is actually rejecting the donor tissue. It occurs when the immune system of the transplant recipient rejects the transplanted tissue. Is that clear now? So just remember that graft versus host disease is that the graft, that is the donor tissue, it acts against the host or the recipient cells. In rejection case, the recipient is rejecting the accepted tissue. Okay. Is that clear now? Okay, now let's look the options. So option A is host T cells attacking the graft. Is that true? They are telling that host means recipient attacking against the graft is actually transplant rejection. Okay, so that is not the answer. Option B, donor lymphocytes attacking the recipient tissues. So here donor means the graft. They are attacking against the recipient, that is the host. So graft versus host disease. So obviously this must be the correct answer. The option C is NK cell deficiency, no. Option D, excess erythropoiesis. So that is also not the correct one. So the correct answer is option B, donor lymphocytes attacking the lymphoid tissue. Okay, donor lymphocytes attacking recipient tissue. One more term is the allogenic. In the previous question we discussed about xenogenic transplantation. Xenogenic means we said that it is transplantation of the tissue or cells between individuals of different species. Allogenic means transplantation that is happening within the same species, individuals of the same species. So in humans, like in us, if I'm receiving a bone marrow transplantation, obviously it must be a human. The donor must be a human. So within the individuals of the same species. So here allogenic donor T cells, they recognize the host antigens as foreign. Same thing that we already discussed right now. So that is the graft versus host disease. So this is all about today's discussion. So here's the references. Then I hope this would be a beneficial discussion for your exam preparations. So thanks for watching. Yeah, don't forget to subscribe the channel if you like the video. See you again.